The Lac Operon is an operon required for the transport and metabolism of lactose in Escherichia coli and some other enteric bacteria. It has three adjacent structural genes, lacZ, lacY, and lacA. The genes encode I2-galactosidase, lactose permease, and thiogalactoside transacetylase, respectively. In its natural environment, the Lac Operon allows for the effective digestion of lactose. The lactose permease, which sits in the cytoplasmic membrane, transports lactose into the cell. I2-galactosidase, a cytoplasmic enzyme, subsequently cleaves lactose into glucose and galactose. However, it would be wasteful to produce the enzymes when there is no lactose available or if there is a more preferable energy source available, such as glucose. Gene regulation of the Lac Operon was the first genetic regulatory mechanism to be understood clearly, so it has become a foremost example of prokaryotic gene regulation. It is often discussed in introductory molecular and cellular biology classes at universities for this reason. The Lac Operon uses a two-part control mechanism to ensure that the cell expends energy producing the enzymes encoded by the Lac Operon only when necessary. In the absence of lactose, the Lac Repressor halts production of the enzymes encoded by the Lac Operon. In the presence of glucose, the catabolite activator protein, required for production of the enzymes, remains inactive, and Ecoli shuts down lactose permease to prevent transport of lactose into the cell. This dual control mechanism causes the sequential utilization of glucose and lactose in two distinct growth phases, known as Doxy. Structure of the Lac Operon. The Lac Operon consists of three structural genes, and a promoter, a terminator, regulator, and an operator. The three structural genes are, lacZ, lacY, and lacA. lacZ encodes I2-galactosidase, an intracellular enzyme that cleaves the disaccharide lactose into glucose and galactose. lacY encodes I2-galactoside permease, an inner membrane-bound importer that pumps lactose into the cell using a proton gradient. lacA encodes I2-galactoside transacetylase, an enzyme that transfers an acetyl group from acetyl-CoA to I2-galactosides. Only lacZ and lacY appear to be necessary for lactose catabolism. Genetic nomenclature: three-letter abbreviations are used to describe phenotypes in bacteria including E. coli. Examples include Lac, His, Mo, SMR. In the case of Lac, wild-type cells are Lac+ and are able to use lactose as a carbon and energy source, while Lac mutant derivatives cannot use lactose. The same three letters are typically used to label the genes involved in a particular phenotype, where each different gene is additionally distinguished by an extra letter. The Lac genes encoding enzymes are lacZ, lacY, and lacA. The fourth Lac gene is lacI, encoding the lactose repressor. I stands for inducibility. One may distinguish between structural genes encoding enzymes, and regulatory genes encoding proteins that affect gene expression. Current usage expands the phenotypic nomenclature to apply to proteins, thus, lacZ is the protein product of the lacZ gene, I2-galactosidase. Various short sequences that are not genes also affect gene expression, including the Lac promoter, lacP, and the Lac operator, lacO. Although it is not strictly standard usage, mutations affecting lacO are referred to as lacOc, for historical reasons. Regulation: specific control of the Lac genes depends on the availability of the substrate lactose to the bacterium. The proteins are not produced by the bacterium when lactose is unavailable as a carbon source. The Lac genes are organized into an operon. That is, they are oriented in the same direction immediately adjacent on the chromosome and are co-transcribed into a single polycistronic mRNA molecule. Transcription of all genes starts with the binding of the enzyme RNA polymerase, a DNA-binding protein, which binds to a specific DNA-binding site, the promoter, immediately upstream of the genes. Binding of RNA polymerase to the promoter is aided by the compound catabolite activator protein. From this position RNAP proceeds to transcribe all three genes into mRNA. The DNA sequence of the E. coli Lac Operon, the lacZR mRNA, and the lacI genes are available from GenBank. The first control mechanism is the regulatory response to lactose, which uses an intracellular regulatory protein called the lactose repressor to hinder production of I2-galactosidase in the absence of lactose. The lacY gene coding for the repressor lies nearby the Lac Operon and is always expressed. If lactose is missing from the growth medium, the repressor binds very tightly to a short DNA sequence just downstream of the promoter near the beginning of lacZ called the Lac operator. The repressor binding to the operator interferes with binding of RNAP to the promoter, and therefore mRNA encoding lacZ and lacY is only made at very low levels. When cells are grown in the presence of lactose, however, a lactose metabolite called allolactose, which is a combination of glucose and galactose, binds to the repressor, causing a change in its shape. Thus altered, the repressor is unable to bind to the operator, allowing RNAP to transcribe the lac genes and thereby leading to higher levels of the encoded proteins. The second control mechanism is a response to glucose, which uses the catabolite activator protein to greatly increase production of I2-galactosidase in the absence of glucose. Cyclic adenosine monophosphate is a signal molecule whose prevalence is inversely proportional to that of glucose. It binds to the CAP, which in turn allows the CAP to bind to the CAP binding site, which assists the RNAP in binding to the DNA. In the absence of glucose, the CAMP concentration is high and binding of CAP CAMP to the DNA significantly increases the production of I2-galactosidase, enabling the cell to hydrolyze lactose and release galactose and glucose. More recently, inducer exclusion was shown to block expression of the Lac Operon when glucose is present. Glucose is transported into the cell by the PEP-dependent phosphorotransferase system. The phosphate group of phosphoenol pyruvate is transferred via a phosphorylation cascade consisting of the general PTS proteins HPR and EIA and the glucose-specific PTS proteins EIGALC and EIGALC, the cytoplasmic domain of the EII glucose transporter. Transport of glucose is accompanied by its phosphorylation by EIGALC, draining the phosphate group from the other PTS proteins, including EIGALC. The unphosphorylated form of EIGALC binds to the lac permease and prevents it from bringing lactose into the cell. Therefore, if both glucose and lactose are present, the transport of glucose blocks the transport of the inducer of the Lac Operon. Multimeric nature of the repressor. The Lac Repressor is a tetramer of identical subunits. Each subunit contains a helix-turn helix motif capable of binding to DNA. The operator site where repressor binds is a DNA sequence with inverted repeat symmetry. The two DNA half sites of the operator together bind to two of the subunits of the tetrameric repressor. Although the other two subunits of repressor are not doing anything in this model, this property was not understood for many years. Eventually it was discovered that two additional operators are involved in lac regulation. One lies in the end of the lacY gene and the other is about 400 BP downstream in the early part of lacZ. These two sites were not found in the early work because they have redundant functions and individual mutations do not affect repression very much. Single mutations to either O2 or O3 have only 2 to 3-fold effects. However, their importance is demonstrated by the fact that a double mutant defective in both O2 and O3 is dramatically de-repressed. In the current model, repressor is bound simultaneously to both the main operator O1 and to either O2 or O3. The intervening DNA loops out from the complex. The redundant nature of the two minor operators suggests that it is not a specific looped complex that is important. One idea is that the system works through tethering. If bound repressor releases from O1 momentarily, binding to a minor operator keeps it in the vicinity, so that it may rebind quickly. This would increase the affinity of repressor for O1. Mechanism of induction. The repressor is an allostric protein, that is it can assume either one of two slightly different shapes, which are in equilibrium with each other. In one form the repressor will bind to the operator DNA with high specificity, and in the other form it has lost its specificity. According to the classical model of induction, binding of the inducer either allolactose or IPTG, to the repressor affects the distribution of repressor between the two shapes. Thus, Repressor with inducer bound is stabilized in the non-DNA binding conformation. However, this simple model cannot be the whole story, because repressor is bound quite stably to DNA, yet it is released rapidly by addition of inducer. Therefore it seems clear that repressor can also bind inducer while still bound to DNA. It is still not entirely known what the exact mechanism of binding is. Lactose analogues. A number of lactose derivatives or analogues have been described that are useful for work with the Lac Operon. These compounds are mainly substituted galactosides, where the glucose moiety of lactose is replaced by another chemical group. Isopropyl I2D thiogalactoside is frequently used as an inducer of the Lac Operon for physiological work. 1. IPTG binds to repressor and inactivates it, but is not a substrate for I2-galactosidase. One advantage of IPTG for in vivo studies is that since it cannot be metabolized by E. coli, its concentration remains constant and the rate of expression of lacPO control genes is not a variable in the experiment. IPTG intake is dependent on the action of lactose permease in P. fluorescens, but not in E. coli. Phenyl I2D galactose is a substrate for I2-galactosidase, but does not inactivate repressor and so is not an inducer. Since wild-type cells produce very little I2-galactosidase, they cannot grow on phenylgal as a carbon and energy source. Mutants lacking repressor are able to grow on phenylgal. Thus, minimal medium containing only phenylgal as a source of carbon and energy is selective for repressor mutants and operator mutants. If 108 cells of a wild-type strain are plated on agar plates containing phenylgal, the rare colonies which grow are mainly spontaneous mutants affecting the repressor. The relative distribution of repressor and operator mutants is affected by the target size. Since the lacY gene encoding repressor is about 50 times larger than the operator, repressor mutants predominate in the selection. Other compounds serve as colorful indicators of I2-galactosidase activity. ONPG is cleaved to produce the intensely yellow compound, orthonitrophenol, and is commonly used as a substrate for assay of I2-galactosidase in vitro. Colonies that produce I2-galactosidase are turned blue by X-gal. Allolactose is an isomer of lactose and is the inducer of the Lac Operon. Lactose is galactose, I2-14, glucose, whereas allolactose is galactose, I2-16, glucose. Lactose is converted to allolactose by I2-galactosidase in an alternative reaction to the hydrolytic one. A physiological experiment which demonstrates the role of lacZ in production of the true inducer in E. coli cells is the observation that a null mutant of lacZ can still produce lacY permease when grown with IPTG but not when grown with lactose. The explanation is that processing of lactose to allolactose is needed to produce the inducer inside the cell. Development of the classic model. The experimental microorganism used by Francis Section Jacob and Jacques Monod was the common laboratory bacterium, E. coli, but many of the basic regulatory concepts that were discovered by Jacob and Monod are fundamental to cellular regulation in all organisms. The key idea is that proteins are not synthesized when they are not needed. E. coli conserves cellular resources and energy by not making the 3 lac proteins when there is no need to metabolize lactose, such as when other sugars like glucose are available. The following section discusses how E. coli controls certain genes in response to metabolic needs. During World War II, Monod was testing the effects of combinations of sugars as nutrient sources for E. coli and B. subtilis. Monod was following up on similar studies that had been conducted by other scientists with bacteria and yeast. He found that bacteria grown with 2 different sugars often displayed 2 phases of growth. For example, if glucose and lactose were both provided, glucose was metabolized first and then lactose. Lactose was not metabolized during the first part of the dioic growth curve because I2-galactosidase was not made when both glucose and lactose were present in the medium. Monod named this phenomenon Doxy. Monod then focused his attention on the induction of I2-galactosidase formation that occurred when lactose was the sole sugar in the culture medium. Classification of regulatory mutants. A conceptual breakthrough of Jacob and Monod was to recognize the distinction between regulatory substances and sites where they act to change gene expression. A former soldier, Jacob used the analogy of a bomber that would release its lethal cargo upon receipt of a special radio transmission or signal. A working system requires both a ground transmitter and a receiver in the airplane. Now, suppose that the usual transmitter is broken. This system can be made to work by introduction of a second, functional transmitter. In contrast, he said, consider a bomber with a defective receiver. The behavior of this bomber cannot be changed by introduction of a second, functional aeroplane. To analyze regulatory mutants of the Lac Operon, Jacob developed a system by which a second copy of the lac genes could be introduced into a single cell. A culture of such bacteria, which are diploid for the lac genes but otherwise normal, is then tested for the regulatory phenotype. In particular, it is determined whether lacZ and lacY are made even in the absence of IPTG. This experiment, in which genes or gene clusters are tested pairwise, is called a complementation test. This test is illustrated in the figure. First, certain haploid states are shown. Panel shows repression, shows induction by IPTG, and show the effect of a mutation to the lacY gene or to the operator, respectively. In panel the complementation test for repressor is shown. If one copy of the lac genes carries a mutation in lacI, but the second copy is wild-type for lacI, the resulting phenotype is normal but lacZ is expressed when exposed to inducer IPTG. Mutations affecting repressor are said to be recessive to wild-type, and this is explained by the fact that repressor is a small protein which can diffuse in the cell. The copy of the Lac Operon adjacent to the defective lacI gene is effectively shut off by protein produced from the second copy of lacI. If the same experiment is carried out using an operator mutation, a different result is obtained. The phenotype of a cell carrying one mutant and one wild-type operator site is that lacZ and lacY are produced even in the absence of the inducer IPTG. Because the damaged operator site does not permit binding of the repressor to inhibit transcription of the structural genes. The operator mutation is dominant. When the operator site where repressor must bind is damaged by mutation, the presence of a second functional site in the same cell makes no difference to expression of genes controlled by the mutant site. A more sophisticated version of this experiment uses marked operons to distinguish between the two copies of the lac genes and show that the unregulated structural gene, S, is the one, S, next to the mutant operator. For example, suppose that one copy is marked by a mutation inactivating lacZ so that it can only produce the lacY protein, while the second copy carries a mutation affecting lacY and can only produce lacZ. In this version, only the copy of the Lac Operon that is adjacent to the mutant operator is expressed without IPTG. We say that the operator mutation is psi-dominant. It is dominant to wild-type but affects only the copy of the operon which is immediately adjacent to it. This explanation is misleading in an important sense, because it proceeds from a description of the experiment and then explains the results in terms of a model. But in fact, it is often true that the model comes first, and an experiment is fashioned specifically to test the model. Jacob and Monod first imagined that there must be a site in DNA with the properties of the operator, and then designed their complementation tests to show this. The dominance of operator mutants also suggests a procedure to select them specifically. If regulatory mutants are selected from a culture of wild type using phenylgal, as described above, operator mutations are rare compared to repressor mutants because the target size is so small. But if instead we start with a strain which carries two copies of the whole lac region, the repressor mutations are not recovered because complementation by the second, wild-type lacI gene confers a wild-type phenotype. In contrast, mutation of one copy of the operator confers a mutant phenotype because it is dominant to the second, wild-type copy. Regulation by cyclic AMP, explanation of Doxy depended on the characterization of additional mutations affecting the lac genes other than those explained by the classical model. Two other genes, CIR and CRP, subsequently were identified that map far from lac, and that, when mutated, result in a decreased level of expression in the presence of IPTG and even in strains of the bacterium lacking the repressor or operator. The discovery of CAMP in E. coli led to the demonstration that mutants defective the CIR gene but not the CRP gene could be restored to full activity by the addition of CAMP to the medium. The CIR gene encodes adenylate cyclase, which produces CAMP. In a CIR mutant, the absence of CAMP makes the expression of the lacZIR genes more than 10 times lower than normal. Addition of CAMP corrects the low lac expression characteristic of CIR mutants. The second gene, CRP, encodes a protein called catabolite activator protein or CAMP receptor protein. The dual regulation causes the lactose metabolism enzymes to be made in small quantities in the presence of both glucose and lactose due to lactose inhibiting lacI from binding to the operator, but at high CAMP concentrations and in the presence of lactose there are high levels of expression. Leaky expression is necessary in order to allow for metabolism of some lactose after the glucose source is expended, but before lac expression is fully activated. In summary, when lactose is absent then there is very little lac enzyme production. When lactose is present but a preferred carbon source is also present then a small amount of enzyme is produced. When glucose is absent, CAP binds to a specific DNA site upstream of the promoter and makes a direct protein-protein interaction with RNAP that facilitates the binding of RNAP to the promoter. The delay between growth phases reflects the time needed to produce sufficient quantities of lactose metabolizing enzymes. First, the CAP regulatory protein has to assemble on the lac promoter, resulting in an increase in the production of lac mRNA. More available copies of the lac mRNA results in the production of significantly more copies of lacZ and lacY. After a delay needed to increase the level of the lactose metabolizing enzymes, the bacteria enter into a new rapid phase of cell growth. The diagram below summarizes these statements. Two puzzles of catabolite repression relate to how CAMP levels are coupled to the presence of glucose, and secondly, why the cells should even bother. After lactose is cleaved it actually forms glucose and galactose. In metabolic terms, lactose is just as good a carbon and energy source as glucose. The CAMP level is related not to intracellular glucose concentration but to the rate of glucose transport, which influences the activity of adenylate cyclase. As to why E. coli works this way, one can only speculate. All enteric bacteria ferment glucose, which suggests they encounter it frequently. It is possible that a small difference in efficiency of transport or metabolism of glucose v. lactose makes it advantageous for cells to regulate the Lac Operon in this way. Used in molecular biology, the lac gene and its derivatives are amenable to use as a reporter gene in a number of bacterial-based selection techniques such as two-hybrid analysis, in which the successful binding of a transcriptional activator to a specific promoter sequence must be determined. In LB plates containing X-gal, the color change from white colonies to a shade of blue corresponds to about 20 to 100 I² galactosidase units, while tetrazolium lactose and McCunkie lactose media have a range of 100 to 1000 units, being most sensitive in the high and low parts of this range respectively. Since McCunkie lactose and tetrazolium lactose media both rely on the products of lactose breakdown, they require the presence of both lacZ and lacY genes. The many lac fusion techniques which include only the lacZ gene are thus suited to the X-gal plates or ONPG liquid broths. See also, blue-white screen, catabolite repression, references. External links, Lac Operon at the U.S. National Library of Medicine. Medical subject headings, Virtual Cell Animation Collection introducing, the Lac Operon, Bozeman Science, staining whole mouse embryos for I² galactosidase activity.